I'm Nicole and I'm a Scientific Fellow at the School of Optometry and Vision Science. And in my research, I'm trying to keep the cornea, the window of the eye, transparent. And you can see in this diagram that the only reason you can see the cornea is from this reflection. And in fact, the cornea is only 0.5 millimetre thick, but it's an amazing tissue. To keep the transparency, the cells and the fibres have to be very regularly arranged, and there are no blood vessels. So to protect the cornea, we have the eyelids, the eyebrow. In the anatomy, we have biochemical defences in the tears, and the epithelium on the surface of the cornea constantly senses for organisms and debris and sloughs off and regenerates, and that sits on a reinforced basement membrane. In fact, the cornea is actually the most innervated part of the whole body, so it is really primed for fast action. But it's not infallible, and when needed, inflammatory cells will stream into the cornea from the surrounding tissue. And if the stimulus is sterile, such as toxin that have just been coughed up by some bacteria, the inflammation is quite regular, but if it's infection, the infection is messy. So this lesion becomes a conglomerate of not only the organisms, but dead cells and inflammatory cells. And in fact, it's the inflammatory cells that do a lot of the damage. And these lead to scarring and permanent vision loss. To treat the infections, we use intense antibiotics. And when the microorganisms actually die, we tend to get more of an inflammatory reaction. So we often will use steroids in the healing process to limit the damage. And we know that it's important, the timing of the use of those steroids. But steroids have been described as like taking a sledgehammer to a brick wall to remove a brick. And in the eye, they have side effects such as increased pressure and long-term effects on the crystalline lens, such as cataracts. In addition, some microbes get excited by steroids, and some just think, oh, I'm going to have a party because there's a decreased immune response. So I'd like you to meet Irene and Marta. Both of these young ladies have had corneal infections. Irene is now blind in her eye, and Marta has lost a bit of vision. You'll see her reflection here is not as bright as the other eye, but she's doing much, much better than Irene. And we do know that some microbes are more pathogenic than others, that it's important that you get treatment early. And I'm afraid if you're over 30, you'll have a worse outcome in infection than if you're younger. But these two ladies their eyes were infected by the same microbe. They're of similar age and background, so clearly there are some other forces at play. And we know a lot about the inflammatory response in corneal infection from animal models. We know that mice that are bred to have a Th2 response, inflammatory response, have better outcomes than those that exhibit a Th1 response. And these responses are directed by messengers called cytokines. But we know that mice are different to humans, and also recently it's been found that sex is an important biological variable in some of these models that hasn't been taken into account in the past. So clinical studies are very important, and for my PhD, I looked at the genetic code of 5 cytokines that have been highlighted from these animal models in 90 patients and 185 healthy controls. And we did this using pyrosequencing here at UNSW. And we found that with 4 out of 5 of those cytokines, we found association, not a bad hit rate, but the significance was fairly mild. And like many other researchers in the genetics of complex diseases, we have moved more towards the diseased tissue. And our diseased tissue is very accessible. We don't use a needle, but the doctors do in the clinic when they diagnose an infection. And they do it with just using anesthetic drops. So for us, collecting some tears or even collecting a swab of the eye surface is very easy and acceptable for doctors, patients, and even ethics committees. And we've found some significant associations, and here at UNSW, we've been at the forefront of some of those findings. And with the advanced technology that we have on campus here, we're able to do multiplex immunoassays and single cell sequencing to be able to analyze these small samples much easier than in the past. So we are really on the path towards personalized medicine in eye infection. We do have some challenges. The eye is in constant opposition to the environment, which means that it's prone to fluctuation. We do collect small samples and they're prone to degradation, which can be important in a busy clinic. But we have got some promising results, and we are in a revolution of omics. So Google have invested in a contact lens to measure glucose in diabetics, and we have a dry eye test using cytokines that is commercially available. And we are lucky to be on the campus of such a great quantum computing boom, which you'll hear about later, and we hope to use those in our modelling of disease in the future. So our potential uses are as a biomarker, as a prognostic tool, and to monitor management, and we hope that that can help us with better use of our current therapy, and also to develop new therapies with less side effects and contraindication than our sledgehammer steroids. So one of the other aspects of our research is that we actively support Irene as a patient advocate for the better management and diagnosis of eye disease, and I feel confident that in the future we'll be able to do better for patients like her. Thank you.